Hello, this is lecture 3.1, Tour of a Cell. In this lecture, you will learn to identify the parts of bacterial, plant, and animal cells and compare the similarities and differences between them. You will also learn to describe the function of the different cellular organelles. In addition, you are going to learn about the fluid mosaic model of cell membranes. It turns out that there are two major categories of cells on Earth. We have cells that are called eukaryotic cells, and we have cells called prokaryotic cells. The prokaryotic cells are the most ancient, and those are associated with bacterial cells. They're very simple compared to eukaryotes. So all living things can either be classified as eukaryotic or prokaryotic. There's actually two groups of prokaryotic cells, bacteria and archaea. And the eukaryotic cells, there's actually quite a few different varieties. We'll go into those in a little bit more detail later. So prokaryotic cells, these are typically smaller than eukaryotic cells. They do not have internal structures that are surrounded by membranes, and they lack a nucleus. So they have DNA that floats in a cytoplasm. They've got a plasma membrane. They often have a cell wall which is thick and helps to protect them from drying out, a capsule, and a flagellum which helps them to move. So this picture here shows you the relationship between prokaryotic and eukaryotic cells. This is the first time that you'll have seen a picture like this during the course. This picture is a visual representation of the evolutionary relationship between all of these organisms. At the base or at the bottom is a single line representing a single unified ancestor. From this ancestor, we have three groups evolve. The first group of the bacteria, and there are many different groups of bacteria. The next group are called the archaeans, and from the archaeans we end up with the eukarya or the eukaryotic cells. And you'll notice if you look at eukarya, there are some groups that you might be familiar with. Animalia, the animals, fungi, plantae. These are things that we'll talk about more later this semester. There are four types of organisms made of eukaryotic cells that you will know. Plants, animals, fungi, and then a group called protists. And protists is basically a grab bag of all other eukaryotic cells. And most of them are single-celled. Some of them are colonial, where multiple cells will live together. But the plants, animals, and fungi are three kingdoms of eukaryotic cells where we've had the separate evolution of multicellularity. That's where you have more than one cell creating a single living organism. So here's a eukaryotic cell. You could think of this as being your standard animal cell. Honestly, though, if you had a look at a microscope slide and you looked at some different cells from a human body, they'd be quite different from each other because they have specialized functions. But this is a broad overview of what you might expect. In the center of the cell is a membrane-bound nucleus that contains DNA in the form of chromatin. There's a rough endoplasmic reticulum that has little beads, which are ribosomes. We have got mitochondria. We've got a plasma membrane. We've got internal structures like filaments and microtubules and centrosomes. There are organs that are involved in packaging and secretions, such as golgi apparatus and vacuoles. There's quite a few different things inside of a eukaryotic cell. This is another eukaryotic cell, but this time it's a plant cell. There are some differences between plant and animal cells, and one of the most major differences is that the plant cell has got a thick cell wall around the plasma membrane, and that cell wall is made of cellulose, which we've talked about last week. Plants also have a structure called a chloroplast that is the photosynthetic organ. They have large structures called vacuoles that store starches and pigments and other chemicals. There are some other differences as well. between plant and animal cells. We won't get into them right now. So this is a useful diagram for you. I'd suggest copying it out for one of your study sheets. This is showing similarities and differences between plant cells and animal cells. Both of them have got mitochondria, which are, let's say at once, the powerhouse of the cell. This is where the cell makes ATP. We've got cell membranes. That's the outer membrane that holds all of the cell's junk in the middle. We have lysosomes, which are small packages that are used to digest things. We've got the Golgi apparatus. We've got RER and SER. That stands for rough endoplasmic reticulum and smooth endoplasmic reticulum. We have a cytoplasm and we have a nucleus. So that's common between both of the plant and animal cells. Animals have got centrioles and very small vesicles, and plants have got a cell wall, chloroplast, and a central storage vesicle. I suggest you memorize that fact. That'll be a question on one of your quizzes or exams for sure. All right, so fluid mosaic model of cell membranes. What on earth is this about? Well, if you recall our lecture on large biological molecules, we discussed that there was a category of lipids called phospholipids. These phospholipids had two long fatty acid tails and a phosphate head. Now, what's special about this is that it creates a molecule that has got two different properties at different sides of the molecule. The phosphate part, which in this picture is represented by red circles, is a hydrophilic head. That's the phosphate part. Hydrophilic means likes water. That means that the head can dissolve in water. It's polar. The fatty acid part, however, is hydrophobic. We call those the hydrophobic tails. And that means that they act like oils. They don't want to blend in water. This creates a bilayer, a double layer of phospholipids. If you take just a whole bunch of phospholipid molecules and you start shaking them, over time they will start to organize their orientation so that the hydrophilic heads are dissolved in water and the tails are pointing towards each other. So this is the outermost layer of every single cell, and this is what your body's made out of as well. Now, it's easy to think that a cell membrane is this static thing. However, the fluid mosaic model is a tested model that demonstrates that it's actually a very movable surface. So the best analogy I can give for you is if you imagine the top surface of a swimming pool that's got leaves and bits of insects floating around on it. They're moving around. The molecules of water are also moving around. So that is actually true for your cell membranes. The molecules are slowly moving and mixing around. The floaters in your phospholipid bilayer luckily aren't insects. Instead, you've got a whole bunch of different sorts of proteins, and these have got all sorts of functions. Some of these proteins are tubes and function like gates, allowing chemicals in and out. There are other proteins that are involved in signaling both within the cell and between cells. Your cells have to communicate with each other. There are proteins that attach to the outer side of the cell called glycoproteins, and your cell can use those glycoproteins as messengers to say, I am a cell that belongs to me, versus, oh no, this is a coronavirus. So you can actually detect self from non-self. And this wonderful cell membrane has got selective permeability, which means that it can choose what comes in and what has to stay out. Our next lecture on osmosis and diffusion will get into that in a little bit more detail. But it's really remarkable to think that your cell membrane and the cell membrane of a bacteria or a fly all have very, very similar structures. So let's go into the structure and function of the major organelles. I'll do this really briefly because you've got a textbook that you can read through as well. So the nucleus, it has a double membrane, so not just one phospholipid membrane, but a double phospholipid membrane. And reaching between the two halves of those membranes, we have nuclear pores. These nuclear pores are quite important. They allow messenger RNA to leave the nucleus and go out into the cell where it can make proteins. The chromatin is DNA mixed up with histone proteins, and the chromatin is found primarily in the nucleolus. That's the center of the nucleus. And DNA typically does not leave the nucleus. The nucleus is attached to the rough endoplasmic reticulum, which is a structure that's involved in the making of proteins. Ribosomes are those little dots that you find on the rough endoplasmic reticulum. They're important in building the cell's proteins, and they're made of RNA. We'll go into some more detail later on their structure and how they're used. Most eukaryotic organelles have got membranes, but inside of the eukaryotic organisms, cells, you have got a whole bunch of membranes that work together as a system. So from the nucleus, we've got the rough endoplasmic reticulum that's involved in protein construction, and that's got lots of ribosomes embedded on the surface. Attached to that is the smooth endoplasmic reticulum. The function of that reticulum is not to make proteins, but to make lipids instead. Lipids and other chemicals that are created by these reticula are packaged into small vesicles, and those vesicles can go towards the Golgi apparatus, which is involved in packaging, repackaging, and modifying the incoming chemicals. Once those chemicals have been modified and packaged appropriately, they can be secreted in small vesicles, and they can be actually allowed to leave the cell. So for example, if you need to secrete some digestive enzymes because you've just eaten some protein and you need to break it down with the protein digesting enzyme. So here's a closer look at the endoplasmic reticulum. It really, while I like to say it makes proteins, it does produce an enormous variety of molecules. But do remember, RER makes proteins and SER makes lipids. Here's the Golgi apparatus. It's really recognizable if you're given a picture of a cell. Look for something that looks like a really ugly stack of pancakes. So you've got incoming vesicles on what's called the cis face, and then you've got outgoing transport vehicles on the transface. The cis face is the one that is closest to the nucleus, and the transface is the one that is closest to the outermost part of the cell. So plants have also got vacuoles. These are membrane-bound organelles. They store pigments. So later this semester, you're going to be getting some red cabbage, and we'll be extracting the anthocyanin pigment from red cabbage to use it to make a pH indicator. When you eat potatoes, the starch in potatoes is being stored in vacuoles in the center of each cell. And if you like your caffeine, like I do, caffeine would be stored in a vacuole in the coffee bean. We'll learn more about chloroplasts later, but for right now, you need to know that this is the center for photosynthesis. These structures are used to make sugar. The cell takes in photons that come in from the sun, and on the membranes inside the chloroplasts, which are called thylakoid membranes, there are special proteins in those membranes, and those proteins capture the energy from photons and use that to create a hydrogen ion gradient, and that hydrogen ion gradient is used to drive the synthesis of ATP, which is the molecule for energy that's used in plants and animals. This is the mitochondria. This is found both in plant cells and in animal cells. We'll have it in both. Like the chloroplast, it has a double membrane. It has an outer membrane and an inner membrane. The inner membrane is studded with lots of little proteins that are involved in the breaking down of water. No, it's not breaking down of water. The use of oxygen to make water, and the byproduct of that process is the production of ATP. We'll get into more detail on that later this semester as well.